Hello everyone. Today we are going to learn about DNA extraction, its purpose and methods. So what is DNA extraction? DNA extraction involves lysing the cell, solubilizing DNA, which is followed by chemical or enzymatic methods to remove macromolecules, lipids, RNA or protein. So, DNA extraction involves many procedures, but most common procedures are organic phenol chloroform method, inorganic proteinase K, and salting out method, and other one is nylon method, which is also known as kit method. Four stages of DNA extraction are: in first step, we breaking the cell open to release the DNA by using overtaxing and grinding method, in which we use detergent also. So this is the gift of cell lysis, in which we separate the cell from DNA because we want DNA sample and we use detergent in this process. Our next step is step 2, in which we separate DNA from proteins and other cellular debris. It means we discard the supernatant from that sample because it's necessary to remove all the cellular debris. So this is a gift of centrifuge of DNA sample. We centrifuge that DNA 2 to 3 times for that cleaning purpose because we want to discard the extra cellular debris. So step 3 is precipitating the DNA with an alcohol. What alcohol we use for that is ethanol or maybe isopropanol, and also it will add carefully because it will tear up our skins or maybe frostbites will appear because it is an ice alcohol. So if there is a lot of DNA, you will see a lot of white precipitating just because of presence of DNA. Our next step is step 4, in which we cleaning the DNA. The DNA sample can now be further purified, cleaned. It is then resuspended in a slightly alkaline buffer and ready to use. Why we use alkaline buffer? Because it will renature the DNA. So this is the gift of purification of DNA, in which we purify our DNA and that DNA will use for further procedures. So our next step is step 5. We do this for our personal satisfaction just because confirming the presence and quality of DNA. In step 5, we use spectrophotometer, which is an instrument and which is used to determine the concentration and purity of DNA sample. Alternatively, gel electrophoresis can be used to show the presence of DNA in our sample and give an indication of its quality. After that, the question is arise, what can this DNA be used for? So this DNA can be used for molecular analysis, including PCR, electrophoresis, sequencing, fingerprinting, and cloning. So this is the overview of our 5 steps of DNA extraction. In first step, we do cell lysis, and second step, we separate DNA from other cellular debris. In third step, we precipitating the DNA, and also in fourth step we just clean the DNA for further purification, and in fifth step we do for our satisfaction because we check the quality and presence of DNA.